pleasure for me to introduce our colloquium speaker today, Professor Brian Collins. He's associate professor in the department here, so I don't really have to tell you much about him. He got his PhD at the University of North Carolina in Chapel Hill in 2009, where he was working under Frank Zurich on synchrotron X-ray studies. Then he went as a postdoc to NC State University, and from there on to NIST in Gaithersburg as an NRC postdoc. He joined WSU in 2014 and was promoted to associate professor in 2020. And Brian, of course, is known to everybody here because he's got a very strong presence in this department. So, for example, he leads the learning assistant program together with Anya, where they hire and train 16 undergrads per semester to help with the Physics 101 group learning activities. He also, of course, leads the RU program over here, together with Anya, which hosts 10 undergrads across each from the US each summer over here. And he also brought the X-ray scattering and diffraction beamline to WSU. It's a famous achievement, and this thing has been online since 2023. This was done together with Professor Leon Moreau from chemistry. He also leads a very successful research program, and his research interests focus on things like molecular nanostructures, self-assembly, organic electronics and device physics, synchrotron X-ray techniques, nano-characterization, resonant X-ray optical physics, and more. And of his many awards and recognitions, I just mentioned that in 2023, he was awarded the WSU College of Arts and Sciences Mid-Career Achievement Award. And he also received a DOE Early Career Award in 2017. He's got strong ties to the synchrotron research that's done in the country. He's the longest serving X-ray scattering proposal review panelist at the National Synchrotron Light Source 2. And this is, for those who don't know it, it's the newest US synchrotron, which was built in 2015. And also, quite notably, he holds the approved program status at Berkeley, at the Berkeley's Labs Advanced Light Source, which provides some exclusive continuous access to X-ray instruments there. And there are only about a dozen groups worldwide that have this status, out of about 1,500 distinct users in 2024. So it's a great pleasure for us to have him in the department, and we're very much looking forward to your talk.
Thanks, Peter. Yeah, it's great to thanks for that great introduction. And I think the last time I spoke at the Colloquium was when I was interviewing for the job here. So it's been a while. It's been more than 10 years. So I'm really excited to tell you about kind of what's happened since, maybe focusing a little bit more on the most recent stuff. But yeah, I still feel a little new, but I guess not so new anymore. Yeah, so I'm going to tell you a little bit about some of those topics that Peter mentioned, exploring molecular devices and their nanostructures within them using resonant X-rays. And so kind of just to introduce you to the kind of materials and devices that we're looking at here. So in 2000, the Nobel Prize was awarded for the discovery that you can make polymers conductive. And so that resulted in a lot of really exciting new technologies. Probably the biggest success story is the OLED display technology now in all your cell phones. And most recently, because these are flexible, that's led to the foldable phones like the Galaxy Fold 7 that's just come out. So that's kind of one success story, but there's a lot of other possible technologies that this class of materials can result in. So organic photovoltaics, so solar panels, which I'll talk about our work in, but also bioelectronics, smart medicine, a lot of really exciting potential ideas are out there as well, like neuromorphic computing. But they pose challenge. This new kind of class materials challenging because it kind of confounds the traditional nanoprobes we've developed for semiconducting materials over the last 60 years that kind of focus on silicon materials, where electron microscopies aren't as useful because we're dealing with radiation sensitive light elements. We have 3 buried structures that's important in these devices. Molecular alignment and crystallinity is important, but they're very low crystallinity materials that gives them their flexible character. And so diffraction techniques aren't always as useful either, as well as a lot of folks who do stuff with polymers or molecular systems will deuterate or other chemically label their materials so that neutrons can see it from neutron scattering or other techniques. But you need big samples for that. And these are thin film nanometer size samples. So that is not useful there either. So my group has tried to fill this need with developing resonant X-ray techniques to look at these structures and materials. So just to give you the mechanistic background here, we have, like, say, carbon atom and the electronic states, and when they bond together in a molecule, they'll hybridize, and we'll get some pi and sigma bonding states, as well as the anti-bonding unfilled states. And what we do is we come in with X-rays that are tuned in energy to a transition in these molecules that promotes, when they absorb one of the electrons from the still atomic-like 1s state up into the anti-bonding states of the molecule. And if we tune the energy just right, we'll get them to actually resonate into those states. And so this gives us elements, first of all, element sensitivity, because only the carbon atom is going to absorb at this ionization potential. But also bond sensitivity of those carbon atoms that we're interacting with. So here's an example of that. We're calling this molecular fingerprint of the molecule. This is at absorbance versus photon energy at about 300 electron volt photons versus your 1 electron volt of optical photons. You can see a step here where that's the photoionization edge of the carbon atoms, or you know the photoelectron coming off. But instead, if we resonate with right at the step into these molecular orbitals, we can actually get really big enhancements in the absorption, very localized in energy that represent different bonds within that molecule. And so that's just the bonding sensitivity, so we can tell different polymers apart, even though they're made of the same elements even. On top of that, we have, this is an electronic transition. So any electronic transition can have a dipole moment associated with it. And so we can use the polarization of the X-ray to tune that to different orientations and give us a measurement of the orientation of the molecules based on that dipole moment. So from this absorption measurement, that we like to parameterize the interaction of the X-ray with the material, and we like to use the index of refraction. And so through absorption, you might know that the imaginary, that's the imaginary part of the index of refraction, right? And so through, if those have taken the NM2, the Kramer-Kronig transformation gives you the real part. So through this one measurement, we can actually measure the entire complex index of refraction across this absorption edge and characterize the molecular interactions that way. So what does this allow? So we can then take this spectroscopy measurement and pair it with a bunch of different nanoprobes. So this is scanning transmission X-ray microscopy, which we've been developing at the ALS. So you come in with these X-rays, you diffractively focus them to a spot, they transmit through your thin film sample into a photomultiplier tube or something. And then you raster that across to get a map or an image of your material. And so this is really cool because what you'll see here, this is a non-resonant X-ray microscope image of a thin film pure polymer film. And you can see the edge of the film there where there's no film and then kind of nothing happening. That's all very smooth. But as soon as you change the energy to a resonance, suddenly you see the edge still there. It's the same spot in the same film, just the energy's changed. Suddenly we can see the nanoscale domains. These are orientational domains within the molecule. So we can tell at every pixel what's the orientation of that molecule. We can also do this with composition using those different spectroscopy of the different molecules and then extract out a composition map versus position on these as well. So that's really cool. So we've been developing that, but one of the limitations is the 30 nanometer resolution limit from the diffraction optics. So we can't quite get to the kind of diffraction limit with this at this point. So we want to get a little bit further. Besides, this doesn't really give you the statistics of it. You want to get a little bit more of the entire, domain distribution and things like that. So we turn toward resonant soft X-ray scattering, which you can see here. We're just coming in with a collimated beam. It's going through the sample and then scattering into an area detector. And the scattering pattern we get here is encoding the nanostructure in a statistical level. Mathematically, the scattering pattern using the Born approximation is a Fourier transform of the real space distribution of structures. And so we can extract that information through that conduits. what's really interesting here is the intensity of the pattern that we get that encodes that structure is sensitive to the difference in the index of refraction between different locations on our film. So if you have more difference, then you're going to get a bigger signal that you can be sensitive to more subtle things. And so we call that the contrast function. And so I plot the contrast function between two different molecules here as a function of photon energy going from 10 EVs up to many thousands of EV. And right at this carbon absorption edge, you can see this red contrast function jumps up several orders of magnitude. And that's again because these molecules look very different because of their different bonding arrangements. And so we can extract then enhance the contrast or enhance the signal, but also vary it by changing the photon energy. So if I zoom in to this contrast function here, you can see the red contrast function changes by 3 orders of magnitude, depending on the photon energy. And if you're looking at vacuum, so this is more like density, contrast from density fluctuations, that changes at a different pattern with as a function of energy. And so we can tune the energy of our scattering photon to focus on different things that might be ordering in our material and then cross-correlate those different aspects of ordering within a material. So for example, here's non-resonant would be more sensitive to like roughness or porosity. And so here's an example of a scattering pattern from that. This is intensity as a function of scattering angle or Q, right, shown here. That's the momentum transfer of the photon. And that's just like the scattering angle, really. So we can do that. We can take the, I've also plotted here 2 pi over Q, which is kind of your Bragg equation to kind of look at what's the size scale that we're looking at different angles. And so you can see we're sensitive from single nanometers on the right to tens of nanometers and hundreds of nanometers to microns. And this intensity versus Q gives us features at different size scales that indicate there's some ordering at those size scales. So if there are peaks, there might be diffraction type events, or they could be more disordered type of structures. If we go to absorption resonance, though, we see that same profile changes. Some features are enhanced and some de-enhanced. And we can use that variation of these feature intensities with energy deconvoluting from using this contrast function to tell us what is the origin of those scattering structures. So this would be molecular domains because the intensity increases with going to resonance, and this intensity decreases at resonance, suggesting that's more of a roughness or a density fluctuation that is causing that feature. We can also use the polarization of the, come on in if you want. The polarization of the X-ray to give us information on the orientation, which I'll talk about in a couple slides. So we've been developing this since I've been at WSU for a while now, focusing on organic electronics, these ion transport type of materials that I'll talk about in a minute. Others have worked on liquid crystalline materials, looking at the orientation correlations in those materials. So here's a non-resonant scattering pattern, intensity versus Q again, very flat and boring. As soon as you go to resonance, all these resonance peaks show up, giving you the orientation organization at the nanoscale of these molecules. Blackwell polymer nanostructures, these are kind of the next generation that Intel is using to do sub 10 nanometer photolithography for the small transistor sizes that they're trying to aim at. Separation membranes that focus on filtration, and even biomaterials, so biomolecules, the structure of those molecules can be potentially investigated with this. And so that's kind of this explosion of different groups and different fields starting to use this really fun technique. Yeah, we just, actually, I'll just mention that we just published this year a review and annual reviews about this if you want to see more. So I want to tell you about 3 different projects that kind of we've been recently working on in my lab that uses and develops resonant scattering. So the first one is going to be organic photovoltaics involving solar cells. And so just to give you a background of that field, organic photovoltaics or OPVs have been progressing really nicely with the efficiencies going over 20% just this year. So some exciting things there because the state-of-the-art panels you'll buy for your house are about 20% efficient. So we're finally getting to the point where this technology is potentially competitive with that. You can see from some of these startup companies that they have a ways to go, but the concept of making square meter per minute is very real in this case on a kind of a, almost like a Kodak, film making apparatus. Actually, some of these startups originally started with a Kodak apparatus to print these things. The way that it works is you have a single layer printing of what you call a bulk heterojunction. So what is this? This is a mixture of a electron donor and electron acceptor molecule to create that PN junction you need for solar harvesting. But the way that these work, you don't want just two slabs and one junction. You want a huge amount of interface, maybe tens of nanometers apart. And so that's tough to do. And so we have this method where you print these things and you try to get the thermodynamics to phase separate them at that level, freeze that morphology in place to generate the charges that you need. Now, of course, all the charges are generated at this donor acceptor interface. And so that's going to be an important place in the device to investigate the dynamics of what's happening. So there's been a lot of material breakthroughs from the chemist side. We don't deal with that. We just buy our materials or collaborate with chemists. We focus more on the nanostructure, okay? And again, using the Resonant X-rays to do that, we found that we need, again, domains on the order of tens of nanometers and high purity of these domains to aid in charge transport and extraction from these devices. And so a recent publication, for example, here showed that when we use this resonant scattering to look at the domain purity, again, the total scattering intensity from these features in the RSOCs, they are proportional to the mean square fluctuation of those domain purity in those domains. Yeah, question.
Can you, how broad is the absorption spectrum relative to like the black body, like the whole black body?
Well, the absorption spectrum that we're focused on is about 30 EV wide. So from like 285 to what is that after that? 320, somewhere around there. We're past all of the resonances.
I think the question is for the device itself, what sort of solar energy is that?
Oh, you're talking about for this technology. I thought you meant for the resonant scattering. Okay. Yeah. So these molecules tend, I don't have a good absorption spectrum to show you, but they tend to be each molecule can absorb, and they are chosen to absorb in different parts of the solar spectrum so that they kind of can potentially actually beat the Shockley-Kayser limit because they have two different band gaps. But they typically are narrower than, say, silicon. So you typically need two of them to be able to span the entire visible spectrum, going from, say, like 400 to, they're pushing to 900 nanometer wavelengths. So, whereas silicon's is at about 1,000 or something like that is where their band gap is.
No ultraviolet, less infrared.
Yeah, a little bit less infrared. The chemists are pushing it further into the infrared in terms of their absorption capabilities. Yeah. That is kind of your standard quantum particle in a box. They're trying to basically increase the delocalization of the electron wave function on the molecule further and further. So it's a larger and larger molecule, and that kind of lowers the energy levels that way. Kind of your standard particle in a box mentality there. So yeah, so we wanted to study this, but we wanted to look more at that donor acceptor interface that happens within these these devices. So just a little bit of the background physics of how this happens, how charge generation happens in these devices. We start with absorption. There's some efficiency, eta of absorption inside a domain or in a molecule. It creates what we call an exciton. So it's just an excited state of the molecule. It's uncharged, but that excited state can FRED or resonant energy transfer between molecules, hop around until they get to an interface. At the interface, there's this emergent state called the charge transfer state, where an electron will hop across the interface, but will still be coulombically bound with the hole on the other side. And so that needs to further separate to generate the free charges, there's charge separated state in the device. And then eventually hop around to get out of the device itself to be collected at the electrodes. Of course, there's this tortuous path to get out because of this blend that we have. And so that there's a lot of loss mechanisms that can happen along the way that we have to be careful of and really want to quantify to figure out, what's the important factors here to generate the most charge. And so in our lab, we've set up a way to basically quantify each of these steps in the charge generation process. One example is called time delay collection field, TCF, which is a laser pump voltage probe measurement. This is what Case Patterson has been working on in my lab, where you generate charge with the laser pulse while you're holding the device at operating conditions. You allow all of this process to happen, so it either separates, the charges separate into charges, or they recombine on the order of nanoseconds. And then before any biomolecular recombination can happen, you extract the charge with a voltage pulse. So that has to happen, between the nanoseconds and microseconds. And so what that gives us is the total generated charge at the interface. So when we put all these together, we can quantify basically the efficiency of each step in a particular cell. And so here's an example for like 6 different cells, all the different losses: absorption losses, exciton losses, charge transfer losses, biomolecular recombination, and then finally how much was actually extracted from the cell. And these are different cells. So we got, we're loading in the cell different amounts of this acceptor molecule from 5% to 50%. And you can see, of course, we're getting more charge extracted as we get to 50% of that extra molecule in there. Yeah.
Your EX1 and CT1, these are continuum states. So there could be many possible decay paths.
They're relatively unknown. These CT states we're talking about, they're not well understood. They're kind of an emergent state that are hard to probe.
You wouldn't be using a single mode laser to hone these.
No. So, but we can see that the big change here is the losses in this charge transfer state that we're affecting. And so the question is, what morphologically is happening at the interface to allow for better charge separation at this point? So there we turn to our X-ray techniques to characterize the nanostructure of these devices. And so what we're going to do is we're going to parameterize this blend as a 3-phase system, where we have a polymer crystal phase, an aggregated acceptor phase. These are fullerenes, actually, is why I can make them balls. And then a mixed interface between the two, characterized by its volume fractions and composition concentration at the mixed phase. And so you can see we basically have, well, 4, but really 3 independent parameters for this model. And so we need, 3 measurements. And so we measure the composition fluctuations through the resonant scattering. We measure the polymer crystal phase through X-ray diffraction. And then a third measurement really is just knowing the concentration that we put into each of the devices. And so we basically can then take all 6 of those devices, measure their concentration fluctuation with RSOx, measure their crystallinity with XRD, and extract out basically those values. Those are the intensities of each of these features as a function of this acceptor concentration. And you can see one's going up, the other one's going down. And then what we're going to do is we're going to apply our 3-phase thermodynamic model to fit the 3 parameters to this data, which there's our fit. It looks pretty nice. And so what we're going to do with the output of this fit are the values of those volume fractions and compositions as a function of our independent variable, the amount of acceptor in the blend. And so that's right here. So these are the 4 variables as a function of fullerene concentration. And we can see initially, for example, we have no fullerene phase. In fact, we just have a mixed phase, whose composition changes gradually up to a miscibility point. And then we have phase separation into a pure fullerene phase after that. Notably, the mixed phase at this interface decreases as we increase the fullerene concentration. So that suggests we want less of that and more of the pure fullerene phase in this case. But we can go more quantitative than that. Okay, so we've already measured quantitatively the charge separation efficiency at the interface, and we've measured quantitatively the amount of mixed phase at that interface. And so we can correlate those 2 together. Thomas Farron did this in his paper just where he graduated, showing that we have a Pearson correlation of greater than 99% between the charge separation efficiency of that CT state and the mixed volume fraction, the mixed phase at the interface there. So that suggests perfect correlation, maybe you could argue that, but it suggests we want sharper interfaces between these domains for increased charge generation. And so those are some of the takeaways from this particular study. Now, we further, investigating, we said that these are getting to be about 20% efficient. And so the question is, how stable are these going to be in the ensuing time frame? And can we scale them up in terms of production? And so Obaid did this nice study where we studied how stable these materials would be. And in this case, what we looked at was this additive. This is a co-solvent. So typically you dissolve these molecules into an ink. It's got a solvent. In this case, it's chloroform. It's very volatile. It just goes away really fast as soon as you print it. So you add in a low volatility co-solvent to, like, basically you have this wet film that can order and organize and phase separate. And that is what controls the phase separation. This is this chloromethylene co-solvent that we use. Very small amounts, like none than 0.5%, then 1%, you get your best performance. And then as soon as you go 0.5% too much, suddenly you get this huge phase separation on the order of microns. This is just a visible light microscope. Then you have no device at all. It's all shorted out. It's horrible. And so that's not good for stability. It's not good for production, large scale production, right? If you have some fluctuations in your ink that you're producing these square meters of, you're going to have whole patches that are all shorted out. This, though, unfortunately, is the standard in the field. Everybody uses this to get the 20% efficiency. And so that's not great for stability. So we tried a different additive called phenylnaphthalene. We replaced the chlorine with a phenyl group. And we get none of that phase separation, which is really cool. Okay, so we still get best performance down here. Eventually, the performance degrades a little bit, but not much. And to spare the details, we found, though, through XRD texture measurements, that the crystal orientation was correlated with this CT state separation efficiency, suggesting that something about the orientation of these molecules is affecting potentially the charge generation in these devices. And so we want to kind of look more now at the orientation of the molecules. And that's what I mentioned before. Through the resonant polarization of the X-ray, we can have access to that. And so that's what we've been kind of focused on, is developing, can we measure orientation of these molecules? And so to kind of continue that story, I want to take you back to where we first found that these were super sensitive to orientation, when we basically were doing our RSOX experiment, and we had these anisotropic scattering patterns show up on our detector. Even though the sample was isotropic, we could rotate the sample and the pattern didn't change. And it turned out is because of this anisotropic interaction of the polarization with the molecules themselves. And so published back when I was a postdoc, we found that this was sensitive to the orientation of the molecules at these donor-acceptor interfaces, which was pretty cool. And we subsequently did another study where we showed that we could change, through processing, we could change the orientation of the molecules with respect to the interface. And as we did that, this short circuit current density, JSC, of the devices could be enhanced by a factor of 2 just by changing that orientation, or so far it's suggested. So that was really cool. We're really excited about that, but that was very qualitative.
Right,
Yeah.
I can just clarify that what you're doing is with that polarized X-ray, you're exciting some molecular
Transition. Yep.
which then scatters in these directions.
Right. Exactly.
And the molecules themselves are random, but the fact that you're probing it that much.
Well, the molecules, in order to get this pattern, the molecules do have to have some pattern of orientation in a regular pattern.
So think about when you rotate the sample, why wouldn't that do the same thing?
Because the order has to be isotropic itself on the global scale. At the local scale, it's anisotropic. So if you're zoomed in to, say, a domain, maybe there's a domain, there's red molecules, and it's surrounded by these blue molecules, you have this molecular orientation that is maybe resonant with the polarization, and this one that is not. And so that will change your contrast in one direction versus scattering in the other direction, and give you these anisotropic scattering patterns. That's what we call the S and P polarized scattering. So the question is, can we be more quantitative about this, right? This was cool, got a lot of press and stuff like that. Can we actually learn more quantitatively about this? And so we need more, we need better optical models to do that, though. Okay, we just don't have really good optical models. So one of the projects we've done here at WCU with Victor Garcia is look at density functional theory calculations to see if we can develop these optical models. We'll need an optical tensor actually to look at orientation, right? So we used transition potential DFT. We found that if you look at the DFT calculation versus the experimental spectroscopy or the absorption spectroscopy, we get pretty good qualitative agreement. It's not great, so some inaccurate amplitudes, but we get hundreds of transitions that occur in this molecule. So we can't refine this model to experiment. It's just too many parameters to refine. So we need to reduce the parameter space of this DFT optical model. And so what Victor did was he developed a clustering algorithm to locate or identify the principal transitions of this spectrum. This is using a kind of an amplitude overlap type of algorithm where we cluster based on overlap of the amplitudes. This is a matrix of those overlaps with different, one overlap with another transition. And so, of course, the diagonal, they all overlap with each other perfectly. And then as you get further away, they overlap with each other less and less. And so we use that to cluster these into these different specific primary clusters, which are shown down here, the result of that clustering algorithm. And then we can represent that same DFT-based tensor with a few parameters that we can then use to refine to an experimental measurement. And so that's what we did here. This is angle-dependent NextAPS measurements, and we've taken this clustered model and refined it to those, and we can get quantitatively accurate reproduction of all of our data. And on top of that, we can also chemically trace all the different molecular orbitals that we are interacting with, depending on which photon energy. For example, this tall one is just the petals, the out exterior petals of this. It's called copper phthalocyanine molecule. This transition is just the metal center. And then this transition up here is the isoindyl. So it's the nitrogen carbon bonds that we're interacting with. So now we can use this as kind of like Raman spectroscopy a little bit in that now we're trying to characterize what each of these peaks, what part of the molecule are we focused on when we're using that X-ray measurement. And so that's really kind of exciting. But we want to use this. How useful is this type of this type of optical model? So that's what Harlan Heilman is doing right now in his PhD research. So he's using what's called X-ray reflectivity. And this is the example, this animation here. This is from my actual postdoc advisor at NIST. He made this. He's really good at a Blender. And so that's what this is going on. So we're doing you're seeing the reflected beam intensity as a function of angle, and you're changing the polarization. And you're doing that for every different energy across the absorption edge. Now, this is typically an underdetermined inverse problem. So there's a lot of possible models that could still represent this data. Harlan, what he did was he fit this data using a transfer matrix E&M model. And the algorithm was just your Monte Carlo markup chain algorithm to fit it. And so here's a non-resonant set of data, the different S&P polarized reflectance. And we get pretty good fits to just a slab model. So here's the substrate and here's the molecule standing on end. But at resonance, it doesn't fit very well. Okay, because resonance is more peculiar, more particular about the orientation of these molecules. So he then measured this in multiple energies across the absorption edge and tied all the energies together, all of the energies and polarizations together with that optical tensor that Victor made. Okay, so now we have all the energies tied together with one parameter, which is the orientation of the molecule across all energies. And when he fits that across all these energies, we get very good fits, both polarizations at every single energy, and we can get a molecular orientation depth profile. So this is the surface of the film that we deposited. It's got some orientation which changes as you go deeper and deeper into the film. We find there's a bulk layer, there's a surface orientation of the molecules, and there's below the surface interfacial orientation right at the surface of the surface of the substrate, really, between the film and the substrate. So this is really cool. So we can see that we can actually get depth profiling and look at, non-destructively, by the way, orientation of the molecules inside multi-layers of these coatings. And so we're shipping this to, or trying to get Samsung to give us some money to look at this for their OLED displays. So yeah, so we're excited about these possibilities with the organic solar cells, but also the orientation aspects that we're still probing. And so Acacia's working on kind of the dynamics that are happening, and Tanner's been working on understanding the morphologies, the systems that are just kind of, with Tanner just kind of getting started there. So with a little bit of time left, I want to give you some brief kind of glimpses at some of these other projects that we've been doing. So this one's more looking at ion transport in these polymeric conductors. So again, some background quickly. These polymers, this one's called PDOT PSS. It's a polymer electrolyte where you have the pole conducting or the conducting polymer PDOT with PSS. PSS is an ionic polymer, so it's actually hydrophilic. It likes water. And you blend these two together, and you get this really cool effect where our collaborator, George Malieris at Cambridge, showed that you can make these devices that are sensitive to neuron action potentials. Okay, so our neurons fire with ions like potassium and sodium, right? And we need to transduce that signal into a signal the computer can read. And so he made these little electrochemical transistors out of this polymeric material and actually put it in someone's brain to test that performance of the polymer sensor versus the performance of a traditional medical device. And you can see the polymer sensor outperformed the sensitivity of the traditional medical device, which is really cool. And so we collaborated together to look at the nanostructure of these materials, and we find that we know at least that the PSS, which is a chain, which is this gray thing, is connected to the PDOT through an ionic kind of connection, right? Because there's a positive and a negative kind of charge there. They create these gel particles in water because the PDOT is hydrophobic, the PSS is hydrophilic, and so you have kind of a core shell thing happening there. When you print them into these devices, these gel particles collapse into these pancake structures. We have PDOT in the middle, surrounded by PSS kind of layer, skin layer on the outside. And we find that through our RSOX measurement that these can be affected by different types of processing. And what's important to note here is that the electrons will transport through the PDOT parts. And so you can enhance the crystallinity of those and get really great hole transport. And ions come in through this side as a gate. And so if you touch it to a neuron, it fires, the ions come down in here, and that increases the conductivity of the holes, and that gives you the sensitivity to the neuron action potential. But the question is, or the problem is that when you enhance the hole transport, the conductivity of this material, it turns out you de-enhance or you ruin the ion transport of this material. And that's we found, we think, because you make these gel particles really dense, and the ions have to go around them, and it just takes too long to get around it. So the question, I'm going to skip some of this for time. The question really becomes, can we streamline these little channels for the ions to go through, make it easier for them to get through the device? Okay, so they don't slow down so much. Here's the ion mobility dying because these get too dense. All right. So we use the idea, or Tamana, actually, my other grad student, used the idea that we know that because PSS is more hydrophilic, its surface energy is such that it likes to collect at the surface when you print these things automatically. We characterize that to be a few nanometers at the surface. We thought maybe we can use this as just a streamlined nanochannel to let the ions go straight through without being caught up in this quagmire here down with the gel particles. So she did this really cool experiment called an electrochromic moving front experiment, where we're trying to figure out what is the speed or mobility of the ions. And so you can't necessarily do this purely electrically because you don't know if you're measuring ion conduction or if you're measuring electron conduction in these materials. So we need a different way to see it. So what we did was we made these devices. Here's the PDOT PSS channel. We dip it in water, okay, actually electrolyte with sodium ions. And we force the ions to only go on the end of the channel by putting an encapsulation layer, so it's water impermeable. And we put a voltage between the electrode and electrolyte and the gold on the other end of the channel to drive the ions into the channel, okay? And what happens is the ions go in and they, I said that the P dot and PSS are joined with an ionic kind of bond. Well, with the ions come in, they replace the P dot. They separate those two out. And the P dot goes from being transparent to blue. And so that's electrochromism. And so wherever the P dot is blue, we know that's where the ions went. Okay, so we use the color to tell us where the ion goes. And so we click on this. Here's an actual video of her doing this. So we can see it turning blue, and if we follow that front, this basically tells us how fast were the ions going. And that is giving us the mobility of the ions separate from the electrons in this material. So we can do a whole bunch of different measurements on one device, different voltages, different trials. We can fit it to this basic E&M simplified model here to get the mobility out. And we get, here's the mobility, 50 trials or something like that, giving us that value, which I made her do this 50 times. I didn't believe it when I first did this, because you look up the mobility of sodium in pure water and it's 20 times slower than this. So I was like, what the heck is going on? That can't be right. I think that electrophoretic mobility, electrophoretic transport is basically as fast as the ion can go. As soon as you put it in the solid state, it's got to go slower. It's got to go around molecules and stuff like that. This was going 20 times faster than in water. So that was really surprising. But I mean, every time she did it, we got the same result. We did it with different ions. We did it in a whole bunch of different ways to try to rule out other things, but we kept getting the same result. We had to make some assumptions about the ion density, but then you can calculate the conductivity of the ions as well. And here's like the record for ion conductivity down in this black dot. We had, there's two limiting possibilities, the density. So I did both, but you see the range of the ion conductivity is way above what the record is for ions in a material ever reported. So that's crazy. There's this thing called superionic transport, which I don't know if that's what's going on here, but this is kind of a new kind of world record for ion transport in any material, which, if they could harvest this for batteries, that'd be really cool. Some of the press really jumped on that. Charging your battery 10 times faster would be very nice. Anyway, so that was really exciting. We showed some control mechanisms of this. I'm going to kind of skip some of this for time, but basically, we found that if we change this PVA encapsulation layer from a hydrophilic, which is used to attract the ions, to a hydrophobic to repel the ions, the electrolyte, then we could turn off this enhanced mobility. So the hydrophilic, this is water contact angle. So, if the water beads up on it, it's really hydrophobic. If it flattens out, then it's hydrophilic. So that's what we're using to see how hydrophilic these materials are. We tried 4 different materials and found that if we go to the hydrophobic PVC, like PVC pipes, we don't get enhanced mobility. But if we put hydrophilic PVA on there, we get this enhanced mobility. Okay, so it's kind of linear in that way. We could also take off that channel completely by doing a special wash where we can remove the top coat of the PSS. When we do that to the PVC, nothing happens. It stays the same mobility. So it doesn't, that's again is consistent with the hydrophobicity of the PVC kind of driving away the ions from that top channel. Whereas the PVA, which was we used to bring it into the channel, if we don't have the channel in the first place, we don't have that enhanced mobility either. So this suggests you need both the hydrophilic encapsulation and the PSS layer present in order to get this enhanced mobility. And so in the end, this means that the encapsulation layer is really gating the ions into this kind of super highway for the ions. And so I'll skip to the other tests that we did to just eliminate other possible reasons why we're getting this. We spent years, eliminating other possibilities because we're like, this can't be right. So once we were convinced that this was actually happening, we wanted to say, can we use this for something interesting, like a chemical sensor? So Tamada came up with this idea to use PMMA gate. So PMMA plexiglass is a hydrophobic material. Okay, but we found that there's a nice simple chemical reaction with UV ozone that can, and I'm going to be bad because I don't have a lot of organic chemistry knowledge, but change this kind of methyl type group to hydroxyl, and it makes it hydrophilic when you apply UV ozone. It's really an oxygen reaction with water in the air. And so we can make PMMA turn hydrophilic
by applying this chemical reaction. Okay, so what should happen? So we should expose our device. Well, first measure the mobility, then expose the device to this chemical reaction, then test the mobility again to see if it increases, right? So we did that. I'm going to skip some of that stuff. This is the important one, the ECMF experiment to measure the mobility. Here's the control. The unexposed one is constant, of course. And then the exposed one for different amounts of time. For about 20 minutes, nothing changes. And then after that, we get a more than doubling of the mobility as we expected. So that's cool. But we're like, well, can we do this a little bit more real time and in situ? And can we do it electrically instead of taking videos and watching this blue thing change? So we developed this electrical version of the ECMF experiment where we're applying an AC voltage where we have these ions and we're just sloshing them back and forth in the channel. And if they're higher mobility, we should get higher currents. And if you do the calculations, it's about proportional to a square root of the mobility. So we should get maybe not as big of a peak, but we should get a peak in AC current when we do this experiment. So here's the AC current versus time. All right. And we can see in this blue trace that here we turn on the AC voltage. Here we turn on the UV. We get some photocurrent there from the UV. And then here we see a peak in the AC current. So that's kind of what we expected. We can see that when we don't have UV, this is the first trial, we don't get a peak. When we do have UV, we do get a peak. And then after we do a second trial UV, we don't get a peak because again, the chemical reaction's done. It got basically the bottom of this PMMA gate, became hydrophilic, and that's only gonna happen once. So you notice that there's a time delay though. So why didn't it happen right away? Well, this is an oxygen reaction, so it's got to make its way through the depth of the PMMA layer, right? So there's some amount of time that takes for that to happen and reach the bottom of the PMMA, turning it hydrophilic, and then turning on this channel. And so, well, then if you have different thicknesses of PMMA, it should be more different delay times, right? And that's exactly what we see. So we varied the PMMA thickness from like 80 nanometers to half a micron, and we get this time delay that is linear and goes through zero at zero thickness, which is cool. So that suggests this is what we're seeing here. Also, we can get a reaction rate. 7.5 angstroms a second is the reaction rate of this particular reaction. This is pretty neat. So we now have like a chemical sensor. We can time chemical reactions. This is happening inside a device. So it's not at the surface, it's inside something. So that's pretty neat. And advanced materials really liked it. So this is like supposed to represent the fast lane or what do they call that? When you have more than one person in your car. HOV lane for the ions to go faster. All right, so I'm kind of out of time, so I'm going to skip the last nanocarrier thing, but that's been a lot of fun too. I'm going to skip that for time. That was what Terry McAfee and Devin Grabner worked on. Really exciting stuff to allow us to measure these nanostructures in aqueous. So we're looking at micelles for drug delivery type of applications. So I'll just summarize that. We've been focusing on interfaces and organic electronics using resonance scattering and developing resonance scattering to look at the interfaces, and in particular to look at the orientation of molecules. This mixed transport being probed by this to look at how we can enhance ion transport and investigate how ion transport works. And then the last one I skipped, which is the solvated micelle molecules. So with that, I'd like to thank my group. A lot of them have graduated now at various places. And thanks, all the funding agencies that have funded our work in the last several years. And thank you guys for your attention.
Thank you very much for this very nice talk. Questions, Brett?
Yeah, I have a question which I'm sure I'm not an expert on, but I know in a lot of places like Javier, things like that, you get tremendous enhancement in fusion because you restricted it to a membrane, you restricted the dimensionality. Is that something that you or others think about here?
That's one of my hypotheses, is that it's due to dimensionality, due to space confinement.
Yeah.
You know, I'm trying to think of ways to test that. But that's kind of one of the hypotheses that I do have.
Steve,
Yeah, I'm sort of curious about the chemical stability of the organic fluid in a
Yeah.
realistic environment.
Uh-huh.
You know, you can have temperatures below zero or maybe up to 158.
Yeah, absolutely.
Fahrenheit.
Yep.
Will it last 20 years, 25 years?
That's a big question.
For these broad ranges?
Right. So generally, what's that's more on the engineering side of encapsulation of these materials. So as long as you encapsulate them, it's okay. They have an engineering standard of being able to survive in an environment that's 80 degrees C with 80% relative humidity under constant illumination with the UV part of the spectrum. And so they've tested these. As long as the encapsulation holds up, they're fine. But it's all about encapsulation. So
Is it the same kind of encapsulation as used with silicon?
Oh, yeah. I mean, so you can use what you use in silicon, but those are usually, that's like usually glass or something heavy.
Right.
So you could use that. But you know, if you want the flexible and fast manufacturing and stuff like that, you're going to want something more flexible, like a peg or some kind of layer. Those aren't as good. So people are trying different things. But right now, yeah, it's going to be just your standard stuff that you use to encapsulate silicon like glass.
Okay.
Yeah.
When you answered Steve's first question, you might have said that the devices could beat the Shockley Kaiser limit. Was that correct?
Yeah, that's the idea. Well, so that limit is based on one band gap, right? So you choose a band gap and use thermodynamics to see, you know, the absorption versus emission of photons, the black body spectrum, and at a specific band gap, you have a certain limit. So for the band gap of silicon, it's like 30% efficiency or something like that. That doesn't apply if you have multiple band gaps. And so there's a lot of folks who are working on multi-junction cells and things like that, where you put you stack different cells together to have different band gaps. And people like right now, you can look it up. NREL, the National Renewable Energy Lab, has the records for different types of cells, and kind of the record ever cell is like 50% efficient. Of course, those are things that is questionable whether you could ever manufacture something like that, because it's like hundreds of layers that they put together in very careful ways. In this material, you're not doing, and you're just putting casting together, you're printing them all together, the two band gaps. And actually they found, and this is something we're working on right now, is that when you add a third or even fourth component with different band gaps, you get even more efficiency. So the question there is, we're getting more efficiency than just an extra band gap would explain. So there's a lot of questions about why adding 3 and 4 molecules actually does that. So we're kind of looking at those the mechanisms of that right now.
Thanks.
from a technological point of view, like a user's point of view, can you compare the the beamline that we have here to maybe what a synchrotron can do, and then maybe what an X-ray laser like a Daisy in Hamburg can do?
Yeah. Yeah, I mean, I'll say that if you wanted to make so there's a lot of different parameters of a beam that are important for different types of experiments you'll want to run. So if you want highly collimated, very coherent source and things like that, you know, the beamline we have on campus isn't going to compete with a synchrotron. So you want to choose the type of experiments that don't need those sorts of things. And so when we do one of our, so we'll do a lot of this diffraction measurements on our materials, which the diffraction peaks are broad. And so it's not so big a deal to have a more divergent beam. So we can, we can bring in more of the source X-rays and to focus it in on our sample and things like that. When we do that, we're only about an order of magnitude slower than a synchrotron. It's about 10 times slower, which is great. But other types of experiments you just wouldn't be able to do there. I mean, you can't change the energy, like you can't do spectroscopy like we do with the resonance part. But structural, there are more standard structural characterization, diffraction, small angle scattering that's non-resonant, you can definitely do. It also has other things. It's got imaging. So if you have a heterogeneous material and you want to put the beam in different little grains, say a geologist got different grains and they want to say, what's this grain and that grain? You can image it first and then put the beam in those spots.
It's got a bunch of cool features that you might not have in a synchrotron, or you have to find a specific
I see.
beamline.
Okay. Any last questions? Otherwise, let's thank you again. Thank you.